Hi, everybody. I'm going to be discussing a short paper called Protein Stability Promotes Evolvability. And that title pretty much summarizes the hypothesis that the more stable a protein is, the more likely it will be able to tolerate random evolved mutations. So the authors experimentally validated their theory using a simple model lattice protein that binds to small rigid ligands. They compared two nearly identical versions of this protein. One of them they coined the original parent, and that's shown in figure A, and the other was a slightly more stable version termed the stabilized parent. So to investigate the effect of random mutations on stability and binding affinity, they used error-prone PCR on both variants to generate a whole bunch of randomized mutants of each. And in figure 1b, directly from the paper, they showed the number of mutants from each parent that had improved binding ability for 4 different ligands. These ligands can be seen on the x-axis. And it is obvious from this figure that the mutants from the stabilized parent were greatly improved binders, while many less mutants from the original parent had improved binding ability. Also directly from the paper, figure 1c shows free energy of folding distributions for the mutants to the original parent and the stabilized parent. And it's clear from this figure that many more mutations had a destabilizing effect on the original parent compared to the stabilized parent, which had a much more negative delta G of folding to begin with. So this is reasonable. So they also performed a similar experiment with cytochrome P450BM3. The original parent was the 21B3 variant, and the stabilized parent was the 5H6 variant. Figure 3A, directly from the paper on the left of the slide, shows that far more mutants from the stabilized parent had improved target binding compared to those from the original parent. And figure 3b compares the thermal inactivation temperatures of the improved mutants. Because the stabilized parent had a substantially greater T50, it could better endure thermally destabilizing mutations without losing its activity or its ability to fold properly in comparison to the original parent. And obviously, through evolution, novel functions are conferred. So the fact that a more stabilized protein is able to take on more of these randomized mutations, this also means the more likely that stabilized protein will be to take on some important novel function that comes along with one of the random mutations. So this is a pretty cool finding, and I definitely recommend taking a read of this paper.